So today we'll be discussing on the chemistry of the cancer. So mainly we'll discuss what is the cancer and what is the different type of cancer and how it is related to the cell cycle. So today content will be like definition, cancer terminology, cell cycle and its regulations, and there is one gene known as a P53 which act as a guardian for the genome and the types of cancer. So we'll discuss all this topic today. So before that, you can see there is a different terminology in the cancer. So we'll go one by one. So the cancer, it is a group of diseases characterized by uncontrolled cell divisions leading to the growth of abnormal tissue. So in the cancer, you can see the term known as the uncontrolled cell divisions. Okay. So in the cancer, there is uncontrolled cell divisions, and finally we'll have the growth of the abnormal tissue. Another term is the neoplasia. Neo means new. Okay, there is development of new growth. So that is a sudden and abnormal growth in the tissue or organ, and such growth is known as the neoplasm, or you can say it is a tumor. Another term is carcinogen. So it is any substance or agent that promotes the cancer, that is known as the carcinogens. Okay, so agent which causes the cancer, simply you can known as the carcinogens. So these carcinogens are also often but not necessarily mutagens or teratogens. Then another term is mutation, that already I think you have studied in the mutation class. It is a permanent or transmissible changes to genetic materials. So simply we can say there is an alternation in the nucleotide sequence of the genome. Alteration in nucleotide sequence of genome is known as the mutations, that either it might be in the DNA or RNA. Another term is the mutagens. So it is an agent that changes the genetic informations, usually DNA, of an organisms and increases the number of mutations. Okay, so this mutagens causes the mutations by changing the genetic informations. Okay, so these are agents which can change the genetic informations and finally lead to the mutation, and that is known as the mutagens. Another term, that is apoptosis. Apoptosis means the programmed cell death. So in our body will program the death of the cells. If it is a old cells or it is a damaged cells, it undergoes the apoptosis. Another term is the metastasis. So this term is used, you can say, the spread of the cancer from its primary state to the other part in the body. Like the cancer is present in one organ, which can be travel to the another organs, which can be spread to the other organ, and that is known as the metastasis. Like it is in the liver, through the blood it may reach to the some other part of the body, that is known as the metastasis. There is some gene like the oncogene, proto-oncogene, tumor suppressor genes. So what is the oncogene? It is a gene that can causes a cells to develop into the tumor cells. Okay, the normal cells, but due to these oncogenes, the cells can be changed into the tumor cells and finally result into the cancers. Okay, so normal cells, due to these oncogenes, causes the develop into the tumor cells and finally result into the cancer. Proto-oncogene, it is a gene that is involved in the signal transductions, execution of the mitogenic signals, usually through the protein products. Okay, the product which is formed through that. Often the activation of these proto-oncogenes directly or its products become a tumor inducing agents, or you can say the oncogenes. Like it's due to some changes, these proto-oncogenes can be changed into the oncogenes and lead to the cancers. Tumor suppression is by name itself known as the suppressors, which can suppress the tumor development. It is a gene that reduces the probability that a cell in a multicellular organisms will turn into the tumor cells. Okay, so it prevents the development of tumor. Some example you can say P53 RB genes. These are the genes which suppresses the development of tumor, and that type of gene is known as the tumor suppressor genes. So here you can see oncogenes, which causes the cancers, which changes the normal cell into the tumor cells and finally result in the cancer, and this tumor suppressor genes which reduces the tumor development. Okay, an example is P53 and RBG. Now come to the cancer. As I told before, it is a uncontrolled cell divisions and finally lead to the growth of abnormal tissue. That is known as the cancer. It is a group of disease characterized by the uncontrolled cell divisions and leading to the growth of abnormal tissue, and that is known as the cancer. But if you see that normally in our body, proliferation of the body cells under the strict control. It's under the strict control, the cell proliferations. Okay, and the cells can be differentiate. It will be dividing and the die in the sequential manners. In the sequential manner, it will be differentiate, divide, and the die. This will happen in the sequential manner in the normal conditions. Okay, in the normal conditions. But in the case of cancer, there is a loss of control of cellular growth and development. Just remember, loss of control of the cellular growth and the development, and finally lead to the excessive proliferations of the cells, and that can be spread to the other tissue. But normally it is under the control, but in the cancer cells it is under the uncontrolled. Loss of control will be present in the cancerous cells, so it can divide without any control. There will be no control. There will be no control on the dividing and in the dying of the cells, and that finally lead to the abnormal growth of the tissue. So just like a corona, how it speaks nowadays, similarly the cancer also speaks. Cancer says, I am the world's second largest killer, characterized by uncontrolled proliferation of the cell, implicating environmental and the genetic factors, perhaps due to the imbalance between oncogene and the anti-oncogenes. Imbalance between these two, oncogene and anti-oncogenes. Just to remember, second largest. The first is the nowadays it is cardiovascular disease, which is the first largest killer. The second, the cancer, characterized by uncontrolled proliferations. It is due to the environmental and genetic factors, and also imbalance between the oncogene and anti-oncogenes. Okay. So this is regarding the definitions of the cancer. Now we'll discuss what is the cell cycle and how it can be regulated, because it totally depends on the related to the cancers. So we'll discuss what is the different phases of cell cycle and how it is regulated. What is the different checkpoints that regulates the cell cycles. And I think already you have studied in the +2. So here you can see the first, what is the first gap phase or G1 phase. You can say this one, this blue color, this is a G1 phase. This is the first one. Okay, and this totally depends on the cell type and the developmental stages. Okay, it depends on this cell type and developmental stage. That is the G1 phase. Then you can next one, this one is the S phase. This is another the synthesis phase where DNA synthesis occur, and it complete in the 6 hours. Okay, it will take around 6 hours to complete. The synthesis will occur here, followed by the G2 phase, this one, which takes around the 4 hours to complete. 6 hours for the S phase and 4 hours to complete the G2 phase. Then you have the M phase and with the mitosis phase, it will take only 1 hour to complete. Okay, out of all this phase, like G1 phase, S1 phase, G2 phase, and M phase, we'll have the one more, that is the G naught phase. Okay, G naught phase, where the resting cell, undividing cell will be present here. Okay, undividing cell will be present in the G naught phase. Okay, when it is not dividing, when the cell cycle is just off, that time it will present here in the G naught phase. And in this you can say there is some checkpoints like G1 checkpoints between the G1 and S phase, and another checkpoint between the G2 and M phase. So this is to control the cell cycles. You can say the restriction point or the checkpoint where it can restrict or it can stop the cell cycle. So by checking these checkpoints or stopping these checkpoints. Okay, so totally it may takes around 20 to 22 hours to complete the cell cycles. Okay, this all G1 phase, S phase, G2 phase, M phase, it will take around 20 to 22 hours. So these are the some proteins which help in the regulation of these cell cycles. Like this is a cyclin. Okay, cyclin, the proteins, and this is the CDK. This is cyclin dependent kinase. Cyclin dependent kinase. This is the enzymes, and this is the cyclins. Which both will combine together. Okay, and what is the phosphorylated? It will be active, and this complex is known as cyclic dependent kinase cyclin complex. Cyclin dependent kinase cyclin complex, when it is phosphorylated, it will be active. In this part you can see there is an inhibitors which is presented. This is inhibitors. So inhibitor, if bind to this complex, it will become the inactive, which is also produced after the completion of the cell cycle. When you don't need this cell to divide, that time the inhibitor will be synthesized by the tumor suppressor genes. That will help to synthesize the inhibitors. It will bind to this complex and it will be inactive. So what is the function of this cyclin and cyclin dependent kinase? What it will do to the cell cycles? As you can see here, like this, cyclin A related to the S phase, cyclin B is related to the G2 phase in the G2 phase, cyclin D is related to the G1 phase, and cyclin C, G1 and S phase. Like this is related. So what are the different function of this cyclins? So we'll discuss here in the next slide. It is a protein involved in the cell cycle regulations. Okay, protein which involved in the cell cycle regulations, and it's classified based on the different phase where it is utilized. Based on that, the cyclin are classified or divided. You can say S phase cyclin, example is cyclin A. G phase cyclin, like cyclin D, C, E, and the F. And M phase cyclin is cyclin B, as I have shown in the figure here, like this. So it is which phase it is related, based on that it is known as the, it is classified. So what is the function of all this? So cyclic dependent kinase, by the name itself you can consider enzymes. This is another proteins which involved in the regulation of the cell division cycles, and there is a different types. You can say CDK 1, 2, 3, 4, 5, like that. Okay, and this also classified based on the phases of the cell cycle. S phase CDKs, G phase, and M phase CDKs. That is a classification of the cyclic dependent kinase, and even the cyclins that also differ classified based on the where it is function or utilized, based on S phase, G phase, or M phase cycling and the CDKs. So there is one term that is known as the cell cycle engines. Okay, cell cycle engines, or you can say the complex. That is like cycling dependent kinase cycling complex. Okay, this is known as the cell cycle engine, cyclin dependent kinase cyclin complex. So this consists of the two subunits. One is the cyclin dependent kinase, another is the cyclin. So in the absence of the cyclin, this cyclin dependent kinase are inactive, and there is no kinase activities. That lacks the kinase activities. So when it's combined with the cyclin dependent kinase combined with the cyclins, it forms the complex. and then it will becomes the active. Okay, it becomes the active, as you have seen before in the diagram. Is the diagram like this. These two will combine together and it forms the complex cyclin dependent kinase cyclin complex, which is phosphorylated and it is active. Okay, it becomes the active. So what is the function of that? This CDK in the cell cycle. What is the function? Cyclin dependent kinase cyclin complex. What is the function of that in the cell cycle? Facilitate the passage of cell through various phases. This is the one function. Facilitate the passage of cell through the various phases like G1 phase, S phase, G2 phase, and M phase, and also the checkpoint of the cell cycles. It also has to cross the checkpoint of the cell cycle like G1 checkpoint and G2 checkpoints. Here you can see CDK2 complex with the S phase cycling. CDK2 cyclic dependent kinase 2 complex with the S phase cyclin induces the S phase. Yes, induces the S phase, and thus also help in the crosses of these checkpoints. G1 S checkpoints also will be crossed by combination of these two, cyclin dependent kinase 2, which combined with the S phase cyclin. Second example you can see here, CDK1, the cyclic dependent kinase 1 complex with the M phase cyclin, which induces the M phase and also help to crossing the second checkpoints, G2 M checkpoints of the cell cycles. So this is the function of this complex cyclic dependent kinase cyclin complex, which help in the passes of the cell through the various phases and the checkpoints. Here you can see like this. Okay, this complex induces the and help in the passes to the different checkpoints. And as I told you, the others cyclic dependent kinase which require for the transitions from G0 to G1 phase. So how it is activated, you can see like cyclic dependent kinase 1 and cyclic dependent kinase 2, how it is activated. So here the phosphatase which removes the phosphate. Okay, there is two phosphate here, you can see this phosphate and this phosphate. So this phosphatase will remove the phosphate group from the CDK1 which combined with the cyclin. Now it is inactive form. When this phosphate will remove the phosphate group, one phosphate group, then it becomes the active. Now CDK1 plus cyclin, this complex which is phosphorylated, now it is active. Similarly, you can see this side, cyclic dependent kinase 2 and the cyclin which is inactive, okay, by the kinase enzyme which attaches the phosphate group. Okay, it is phosphorylated and it becomes the active. Now this can help in the passage of cell through the different phases like cyclic dependent kinase 1 and the cyclic dependent kinase 2 which complex with the cyclin. So in this way it will be activated. One, by removing the phosphate, second by adding the phosphate. So finally what you can say, in the phosphorylated state this both will be active. Okay, here on phosphate group here. So in this way this cyclic dependent kinase cyclin complex will be active. There is some non-cycling protein which also help in the cell cycles. So one of the example is the P53, product of the tumor suppressor gene, which is a non-cycling protein. Non-cycling proteins. Till now we have discussed only the cyclin proteins and the cyclic dependent kinase and its complex. But there is one more non-cyclic protein which help in the cell cycle, that is known as the P53. That is a tumor suppressor product of the tumor suppressor gene. What the function of this P53? It acts as a transcription promoter. And activates the gene encoding the CDK inhibitors. As I told you in the first diagram, CDK inhibitors to the complex cyclic dependent kinase cyclin complex inhibitors. Okay, activates the gene which code for the CDK inhibitors. So these inhibitors will be produced and it bind to this complex, and it causes the inactivation of CDK. Okay, as I can show you in the diagram here, like this, CDK inhibitor proteins. So this will come and bind to this complex. Now this is inactive. This will becomes the inactive. So this P53 will activate the gene which code for these proteins, these inhibitor proteins. So this can combine with the complex and it can control the cell cycles. Okay. So what is this P53? It is a 53 kilodalton proteins. It regulates the gene expressions and control the several key genes involved in the growth regulations. It also facilitates the DNA repairs. When DNA damage is encountered, P53 senses that DNA damage and causes G1 arrest of the cell until the damage is repaired. So what do this P53 will do? When there is a DNA damage during the cell divisions, that will be sensed by this P53, and this P53 causes the arrest of the cell cycles. Once there is this damage is repaired, then cell cycle will again resumed. If this damage is not repairable, it is not able to repair, this undergoes the apoptosis. Okay, undergoes the apoptosis, which is not repairable, then undergo the apoptosis. Apoptosis will be triggered by the P53. First is try to repair the DNA damage, the damage which is in the DNA, that is going to repair. If it is not repair, it will activates the apoptosis. You can see here like this. Here there is a DNA damage due to the any cause, you can say. So this P53 will sense it and stop the cell cycles. Okay, once this DNA damage is repaired, then cell cycle will continue. Okay, cell cycle will continue, otherwise this apoptosis will be activated. This triggers the P53 triggers the apoptosis, and that cell will be undergo the programmed cell death. Okay, more simple form you can see here: DNA damage, cell cycle abnormalities. So there is a sense by the P53, cell cycle arrest, DNA repairs, cell cycle restart. Okay, otherwise apoptosis, death and elimination of the damaged cell. This is under the normal conditions. Normally this will happen. In the normal condition this will happen, but not in the cancers. In cancers this control will be omitted. This control is not under these controls. There may be the mutation in this P53 which is not able to sense the DNA damage, and there is no cell arrest. The cell cycle will not arrest and cell will keep on dividing. That will happen in the case of cancer, but normally this will happen when there is any damage in the DNA, that will be sensed by the P53. This is also known as the guardian of the genomes, and arrest the cell cycles. If that damage is not repaired, triggers the apoptosis and causes the programmed cell death of that damaged cells in the normal conditions. In this way it will controls the cell cycles. So this part you have normal, how it happens we have discussed. In the mutated cell you can see mutated P53, which is not sensed by this mutated P53. Finally the cell cycle will continue. Cells can become the cancerous. This will happen in the cancer cells. There is a mutation in P53 which is not able to sense the DNA damage. Okay, and cell cycle will continue, and finally it becomes the cancerous. Now, as I told, this complex will form cyclic dependent kinase, cyclic complex, cyclic complex, which will be active, and cell cycle will start. Okay, but how this will become the inactive. If it is continually working, the cell cycle keep on dividing. Okay, so how it can be inactive? So there is one protein known as ubiquitin. Ubiquitin proteins. Okay, through this ubiquitin pathway, this complex can be becomes inactive. So this ubiquitin go and bind to the cyclin. Okay, and it forms the ubiquitin CDK proteasome. Okay, proteasome. There is a formation of the proteasome when this ubiquitin bind to the this complex, mainly to the cyclins, and there is a degradation of the cyclins. Cyclin will degrade and detach from the cyclin dependent kinase, and now it becomes the inactive. In the starting itself I told, when it is combined with the CDK kinase and cyclin, then only it will be the active form. But the ubiquitin causes degradation of the cyclin. So this cyclin will detach from the cyclin dependent kinase due to the degradation by the ubiquitin, and now this cyclin dependent kinase will become the inactive, and there is arrest of the cell cycles. So in this way, this complex will become the inactive with the help of the ubiquitin proteins. So you can the ubiquitin dependent proteolysis of the cyclin. This is the pathway by that this complex can becomes the inactive, and there is the arrest of the cell cycle. So how this is related to the cancer, the cell cycle gene and the cancer, how it is related? So the gene encoding the cell cycle protein is known as cell cycle genes, and it is designed as the small CDC gene. So gene is generally written in the small form, CDC gene. Okay, so defect or mutation in CDC genes lead to the certain cancers. Just remember, defect or the mutations, both in this gene which code for the cell cycle CDC gene causes the cancer. Defective CDC gene produces defective cell cycle proteins that may causes the cancers. Sometimes the product of CDC gene interact with the p53 proteins, which account for the many cancers. Mutation in the checkpoint genes lead to the cancers. So this is a way how this gene, cell cycle genes related to the cancers. Either there are mutations in the gene or defective products, or mutation in the checkpoint genes, and finally lead to the cancer. So how these proto-oncogenes converted into oncogenes? What are the different mechanism by which these proto-oncogenes, which is generally not the oncogenic? In the normal individuals, this proto-oncogene is not the oncogenic, but once it is converted into the oncogene, it may cause the cancers. So this is a different way by which these proto-oncogenes can be converted into the oncogenes. One is, you can say, the translocation, chromosomal translocation or transpositions, and you can see the normal growth stimulating the protein in excess. Another is the gene amplifications. Here also you can have the excess proteins, normal growth stimulating protein in the excess. Here you can see the point mutations, oncogenes. Okay, point mutations. So here mutation within the control elements will have the mutations. Here also you have the excess proteins. Here within the genes, mutation within the genes. Okay, hyperactivity, degradation, or resistant proteins. Okay, so we'll see how these translocations, what do you mean by this translocation or amplifications, which is increases the protein synthesis. We can see here. So one example for the Burkitt lymphoma, chromosomal translocation in the Burkitt lymphoma, you can see. This is the two chromosome number 8 and chromosome number 14. This here you can see MCOMS which is present in the chromosome number 8, which is transferred here, you can see in the chromosome number 14, transferred here like this, translocation. This part, a small part from this chromosomes, a small part of gene is cut and transferred to the another. Okay, so here it transferred, and that leads to the Burkitt lymphoma. In this way, there is a translocation of the only the some part, not the whole part. Some part of gene will be cut from the one chromosome and transferred to the another, and that leads to the lymphoma or cancer. And this is the example of the Burkitt lymphoma. This is an example of the chromosomal translocation. Gene amplification, you can see normally what will happen. This is a gene. Okay, normally normal protein activity control the cell growth. In normal condition this will happen, but in the abnormal, due to the amplification, see, this is gene, this is gene, this is 3, you can say amplified gene it is. Many more copies of the gene than the normal. Here only one, you can see here. Okay, here you can see 1, 2, 3. So many copies of the gene than normal, and you can see the increase the protein activity, uncontrolled cell growth. So this is happening in the case of the amplification. So in this way, as I told, by the amplification or translocations that converted the proto-oncogene to the oncogenes, and the mutations, as I have told, mutation is also here, which is the main one. So in this way, the oncogenes can be converted into this proto-oncogene converted into the oncogenes by the translocations, gene amplifications, and the mutations by this way, and finally it causes the cancer. So how this cancer is developed, just to take one example, and how it is goes to some other tissue. This is normal cell, mutation in the proto-oncogene or tumor suppressor gene. You can see this one, this is the blue mark, light blue, this one, mutations. So proliferation of the mutated cells. So you can see the more mutated cell is formed, and multiple mutation in the proto-oncogenes, mutation in the tumor suppressor genes. So you can see here, invasion of the surrounding tissue. Before it there was normal, then one mutations which is again proliferated, and you can see more number is increased, which can invade the surrounding tissue. Okay, from the tissue it may reach to the blood. Okay, from the tissue, invasion of the blood, first the surrounding tissue, then it invades the blood, and through the blood it reaches to some other parts, some other organs, and that is only the metastasis, which can spread from one part to another part. Okay, that you can see here how the cancer is developed. Mutation in the proto-oncogenes, then the proliferations, more number is formed, invade the surrounding tissues, then it invades the blood and distribute some other organs and cause the metastasis. So in this way the cancer will be spreading, and there is a formation of the or development of the cancer. So till now this is just the introduction how the cancer is formed when there is uncontrolled in the cell divisions or the cell cycles. So as I told, this cancer is the second largest killer. Okay, in the developed and developing countries both, you can say. So 1 in 4 deaths are due to the cancer. 1 in 4 deaths are due to the cancer, and 1 in 17 deaths are due to the lung cancers. And lung cancer is more common in the male. Similarly, you can say the breast cancer is the most common in the female. And there is many more different type of cancer is present. More than the 100 different form of cancer is present. Okay. So what are the properties of the cancerous cells? Once it is from the normal cell converted into cancerous cell, so what is the properties? It is not under the control, the one diminished, or it is not under the control of the growth. So growth will be not under the control. Capability of invasion of the local tissue. It can invade the local tissue. Cancerous. First one is not under the control. Second, it can invade the local tissue. Capable of spreading to distinct part of the body. Metastasis. So this is the three main properties of cancerous cells. Growth is not under the control, invades the local tissue, and causes the metastasis. That means from one part through the blood reach to the another organs. Okay, this is the major three properties of the cancerous cells. So this cancer, you can say cancer or the tumor. It is any abnormal proliferation of the cells. There is two types. You can say the tumor, benign tumor and malignant tumor. Benign and malignant tumor. So this benign tumor is confined to its original locations. Just note down, it's confined to the original locations. Neither invading surrounding normal tissue nor aspirate to distinct part. Example, you can say the moles. It is confined to the one part. When it is present in the one part, it will not aspirate to the other part. It will not invade to the surrounding tissue. It is confined to the original locations. That's why it is known as the benign tumor. The example is the moles, you can say. Another type is the malignant, characterized by the uncontrolled proliferations, aspirate of the cell to various parts of the body. See, and it can be the cancerous. It is referred to the cancerous. These malignant tumors, which can aspirate to the other parts. The proliferation is uncontrolled, and this is the cancer. Example, you can say the lung cancer, breast cancer, colon cancer. Okay, so this malignant tumor is known as the cancers, not the benign. This benign tumor is not known as the cancer. It is confined to the only one part. It's not invaded to the surrounding tissue, not aspirate to the distinct part. So what is the difference between these tumors, benign tumors and the malignant tumors? Often encapsulated, non-encapsulated, well differentiated, poorly differentiated, low mitotic rate, high mitotic rate, non-invasive, invasive. Metastasis is not present here. Metastasis is present in the malignant tumor. So this malignant tumor is the cancerous. Okay, this is the cancerous, which can aspirate from one part to the another part of the body. So this cancer, depending on the where it is present, what type of cell is involved, based on that you can name it, different name is given, like carcinoma, sarcoma, leukemia. Okay, different types, lymphomas. So the carcinomas around the 90% of the human cancers, and this is malignancy of the epithelial cells, that is known as the carcinomas. Sarcoma is rare in the human, solid tumor of the connective tissue. Okay, connective tissue tumor, that is a sarcoma, like muscle, bone, cartilage and fibrous tissue. Leukemia and lymphomas, it cover only the 8% of the human malignancies, arises from the blood forming cell and the cell of the human systems. Blood forming cell, that is leukemia, and lymphoma, you can say cell of the immune system. So this is a blood forming cell in the cell of immune systems, that known as leukemia and the lymphomas, and sarcoma is the connective tissue, the tumor of the connective tissue, and carcinoma is the malignancy of the epithelium. So based on the cell, which cell is involved, based on this, the name is given. It is a carcinoma, sarcoma, leukemia or lymphoma. Leukemia means blood forming cells, cancer of blood forming cells. So these are some of the oncogenes, their mechanism of activations and the associated tumors. These are the different tumors, and this is the proto-oncogenes. Okay, proto-oncogenes, activation mechanisms, overexpressed or amplified, or there is a translocation or mutations. So this proto-oncogene undergoes these mechanisms, okay, and causes the different type of cancers. So you can take the one or two examples, like here, epidermal growth factors, receptor family, ERB1 and ERB2, overexpression, and say amplifications, like breast cancers, ovarian, lungs, and stomach cancers. Okay, ERB B1 overexpression, squamous cell carcinoma of the lungs. Okay, so by this way, even then you can see these mutations, G proteins. Okay, this proto-oncogene in the RAS undergo the point mutations and cause the multiple cancers including the lungs, colon, thyroid, pancreas and many leukemias. So in this way, these proto-oncogenes by these activation mechanisms converted into the oncogenes and cause the different type of cancers. Okay, so this is, I think, end of the class, and these are few MCQs. You can go through this MCQ, and the next time I will be asking you regarding this MCQ.